Let's talk about Skye Terriers. Let's move on. Oh, yeah, this picture will pop up at various breaks during my talk. This is about half an hour's drive away from where I live. We have many fewer lakes than you have in Finland, but Lake Annecy is the cleanest lake in mainland Europe, which is nice. As you know, liver disease has been a concern of Skye Terriers for some years, as I'll show you. And looking at the UK Skye Terrier Club health report in 2024 and indeed in 2023, it still remains really top of the listed conditions of concern, closely followed by renal dysplasia, and we'll talk a little bit about those two today because it's potentially possible there might be a link. So hepatitis or liver disease in Skye Terriers was actually first reported by Susan Haywood and others in 1988. And I've worked quite closely with Susan Haywood. She was particularly well known for her work on Bedlington Terriers. She's really quite old now and not in great health, but I've been able to share slides with her, and she's an amazing pathologist. So that was a long time ago. She described 9 related Skye Terriers. She didn't explain how they were related, aged between 18 months and 15 years. 2 of them had no clinical signs, but 7 of them had clinical signs, which are typical, as we'll see of the cases we're seeing now. They tended to go off their food. They had fluid in the belly, which we call ascites. They had yellow mucous membranes, jaundice, but only at the end stage. So jaundice wasn't a significant part of this disease until they got to the end stage. 2 of them were asymptomatic, one of which had cirrhosis, which we consider really quite an end-stage liver disease, but it had no symptoms. Now, the problem with this study, it was great to raise awareness of this disease and to suggest that it's maybe different to the disease in other dogs, but there was no description really about the clinical presentation and the outcome and treatment because it was a pathology paper. And I just show you the table from the paper because since then, Skye Terrier liver disease has been called a copper-associated liver disease in various textbooks and papers because there was copper association in the disease, and I don't know if you can see my pointer, but we can see that the worst affected dogs, so group 3 were the worst affected, had more copper. But you can also see that the amount of copper stain was variable, so sometimes it was only 1+, sometimes it was 2+, and the 2 earlier dogs it was 0. And if you bear in mind that a normal, we accept up to 400 micrograms per gram as a normal copper level in dogs, which is a lot more than in humans, and clearly abnormal is over 1,000, and then there's a grey area in the middle. Bedlington Terriers would have, with copper storage disease, would have levels in the 10,000s. So these are increased copper concentrations, no doubt about that, but they're not massively increased. And she described in her original paper that she thought that the copper buildup was as a result of interruption of bile flow rather than being the primary cause of the disease. So that was all a bit mysterious. When I say all went quiet for 15 years, I'm sure there were still cases out there in Bedlingtons, but nobody wrote anything about it. There was nothing in the veterinary literature. Not Bedlington Skyes, I'd never met a Skye Terrier in person, apart from at Crufts. And then we had a dog, before we had the dog come to us, there was one other paper 15 years after Susan Haywood's paper, which was a case that went to Glasgow Vet School. Yvonne McGrossie wrote it up. And this, to me, was quite interesting because this was the first time somebody had described the whole clinical presentation. She had similar clinical signs and pathology. She was a 1-year-old dog, but she actually did very well with treatment, and her treatment was largely supportive. She did have an anti-fibrotic called Chusine, which is of questionable efficacy in dogs, so we don't tend to use it anymore, but the rest of it was supportive. She didn't have any copper chelators, she just had some zinc and ursodeoxycholic acid, which is a biostimulant. The interesting things about her compared with the current dogs that I'm going to tell you about was that her increases in liver enzymes were only mild. She didn't have jaundice, and on ultrasound she had portal hypertension. I'll show you what this is in a minute because this seems to be quite a significant sign in most, if not all, of these affected dogs. And on portal venopathy, which is where you inject a contrast material into the vascular bed in the guts and watch what happens to it as it goes through the liver, they noted that there was abnormal flow on the left-hand side of the liver. Now that is likely to be something that the dog was born with. And so it was normal on the right and abnormal on the left. And again, I will discuss that in more details later. Just to make sure we all know what we're talking about, some of you may know in great detail, and some of you might not, so I just wanted to make sure we're all on the same page. This is my very poor diagram of a liver here. This is the portal vein, and this is the vein that brings all of the blood draining the splanchnic bed. So that's the spleen, the small and large intestine, the stomach, the pancreas. All of it goes through the liver first before it gets into the systemic circulation. And I talk about the liver being like a giant filter paper, basically filtering all of this blood. The blood, I'll show you some microscope slides in a minute, but the vessel splits into multiple parallel circuits effectively, which are lots and lots of little tiny vessels going through the liver, and that's where all the work is done on taking out toxins and taking out food particles, and then they all gather up together again and go into the main vein, and into the main vein here into the heart, which is the caudal vein cava. Now, it's a bit like any flow system. If you start blocking these parallel circuits, a bit like standing on a hose pipe with disease, then you get an increase in back pressure in the portal vein. And that's what we mean by portal hypertension, because as you get more and more fibrosis in the liver, for example, that will block off all the blood vessels, and then the blood literally can't get through. It is like standing on a hose pipe. Now that blood has to go somewhere, so it pools in the spleen, it pools in the intestines, and eventually, if the dog is lucky, and same thing happens in people, they will open up these little shunting vessels that take some of that pressure away. But what this increased pressure does in the meanwhile is it tends to, because the blood is pooling in the guts, it tends to make the guts hypoxic because the blood isn't flowing like it should do, and as a result of lack of oxygen, there are increased risk of getting ulceration. And also it causes fluid build-up because it's literally like a leaky hose pipe, and as the pressure builds up, that fluid squeezes out of the hose pipe into the abdomen. So we believe this is why these dogs show ascites, fluid in the abdomen, because a lot of them have this portal hypertension. And then, as I say, if they're lucky, they get these little escape valves that take some of the pressure away. This was just to show you what I mean by a portal venogram. Nowadays we would do all this if somebody is able to get their dog to a referral centre. We would do this with CT, and we wouldn't have to open the dog up to inject contrast. But here, they've opened the dog's abdomen. There's a little syringe here. They've injected contrast into one of the mesenteric vessels. And what's actually happened? This isn't a Skye Terrier. This was a Sheltie. This dog actually has a complete obstruction of the vein here before it even goes into the liver. So that's given us some useful information. But that's what we mean by portal venography. So the Glasgow dog, rather frustratingly, only had a biopsy of the left side of the liver, the side with the abnormal blood flow. It would have been nice to have compared a biopsy from both sides. And they described, again, changes which are typical of Skye Terrier Hepatitis. We'll talk about this in a minute. And they found some copper on histology, but it was around about only just above the normal range. And they commented about the fact that the lesions were unevenly distributed, which is another feature that we found. In some places the liver is very badly affected, and in some parts of the liver it's less badly affected. And the dog was doing well on its treatment 2 years later when they lost it to follow-up. So then it all went quiet for another 9 years, and this was when we saw our first dog. So it was my first time really getting to know a Skye Terrier, and what wonderful dogs they are. So sort of gentle and quiet and gentlemanly. So this was a male dog, a real gentleman I would call him. So he was an 11-month-old entire male dog who presented with 3 weeks, just 3 weeks of being off his food, being lethargic and having fluid in his belly. On bloods, he had increases in both his liver and his pancreas enzymes, suggesting pancreatitis as well as liver disease. His bilirubin was normal, but his bile acids were high, and they tend to go up when you get shunting through those acquired shunts. So that may be what was causing that. He had a low blood protein, which is another feature of most of these dogs, and normal coagulation times. And actually the owner brought in the article that I've just told you about, which is why I went into it, saying, I would like you to treat my dog like this because this dog did very well. Now, we couldn't go straight away treat without confirming that this was a hepatitis case, because There are many other things that will cause increases in liver enzymes, and they're not all primary liver disease. And so it's important to get a diagnosis. But we got a diagnosis, and in fact, we were suspicious that on presentation this dog had an inflammation of the pancreas, but by the time we took biopsies, the pancreas was looking normal. And again, we gave symptomatic treatment. In this case, because there was a lot of fluid, we gave diuretics. Because he was really a very sick little dog when he presented. He also had blood in his feces, probably as a result of that portal hypertension. So we gave him anti-ulcer medication. And we put him on a high-quality diet, little and often. It wasn't a low-copper diet, it was just a high-quality diet. And actually, I thought this dog was so sick when he came to see us, and he came back having had another relapse a few weeks later. I didn't think he was going to make it. And actually, his symptoms completely resolved, his fluid disappeared, he was a happy little dog, and he was doing really well 7 years later when he was lost to follow-up. And I want you to hold that in your head, because I do think that not all of these dogs do as badly as we think, and some of them actually do really quite well. And I think our view has been perhaps slightly affected by the lack of papers on this, so I'll show you. So this dog made me want to know more, because I had already spent quite a number of years specialising in hepatitis in dogs, and he wasn't behaving quite like a chronic hepatitis. So normally, canine chronic hepatitis cases, they can have a good outlook, but normally they will sort of become slowly progressive, and unless we know the cause and treat a cause specifically, they don't normally do quite so well. They don't normally, chronic hepatitis in dogs, if it presents with a dog that's sick, you would typically have less than 3 or 4 weeks of life left. It implies that the dog has an end-stage disease. So I was a little bit bemused by this. So I was wondering if this was really a typical chronic hepatitis. I was wondering if, I was surprised to see this case, because I think I'd subconsciously thought that this was something that happened in the 1980s and no longer happened. I wanted to know if copper was the cause, and if not, what is the cause? And ultimately, right from the beginning, I sensed the stress about this disease in the breed, and we were hoping that we might be able to find a genetic test for this. Kirsty was saying to me yesterday that would be ideally what everybody would love, and that would be ideally what I would love as well. So this isn't a typical chronic hepatitis, but canine chronic hepatitis is diagnosed on liver biopsies. You can't diagnose it without that. As I said, the signs, the increase in blood samples and so on, do not give you a diagnosis. And what we need to define this disease is a combination of 3 things. We need unhappy liver cells, so this is what this is saying, dead or dying liver cells. So something is attacking the liver. We need an inflammatory cell infiltrate, and we need regeneration and fibrosis. And as we'll see in the Skye disease, we sometimes will call this hepatitis and we sometimes won't, and it really depends on how much inflammation we see. And in most dogs, this is not a very inflammatory disease. For chronic hepatitis in dogs, the causes are often unknown, which is in great contrast to humans. So I wanted to just give you this background and say Skye Terriers are no worse off than other dog breeds, because in the vast majority of dog breeds, we don't know the cause of their hepatitis, and we don't know the genes involved. We know, for example, that Labrador retrievers, about half of them have a copper-associated liver disease. They've identified a gene involved that a lot of breeders use for breeding, but we don't really recommend that, because that gene involved in their copper storage disease only affects about 12% of the heritability of the disease. So unfortunately, although there are breed relationships in chronic hepatitis, the diseases tend to be polygenic, just like hip dysplasia and other difficult to crack diseases. However, some cases are clearly copper, and we can treat them effectively by removing the copper. And some cases, particularly in Springer Spaniels and some Dobermans, are autoimmune, and we can treat them with immune suppression. But we're not really clearly sure that Skyes have either of those, and they certainly don't seem to have autoimmune disease. So, since then, we've been gathering histology blocks from affected Skyes, unaffected Skyes, and we also got the blocks from the Glasgow case and Susan Haywood's original blocks, so we could compare them to see it's the same disease. We've stained with a whole variety of stains, and we've worked now, I've been having Zooms in the last 2 weeks with 2 wonderful pathologists, along with our pathologist at Cambridge, John Cullen, who's the best now dog liver pathologist in the world, and Susan Davis, who's one of the best human liver pathologists in the world. Because we really were trying to understand, can we get any clues as to what the cause of this disease is, and are these all the same disease? And a lot of you will know the late Maud Hawkes. We've worked very closely with Maud, not only at the vet school, but she contributed a lot of samples and energy to the canine genetics group that used to be at the Animal Health Trust, and pedigree information. And as we'll see, we're now all at Cambridge, which makes things easier. So why has it taken so long for us to finally get to a point where we can start trying to unpick what's going on? There's 2 reasons, institutional and the disease. The disease, very difficult to understand. And we now have, since last week in the meeting with the pathologists, I can tell you that this does not look like any human disease. And that's unusual, because most dog and cat liver diseases look similar to a type of human liver disease, but this doesn't. It doesn't really look like any other dog or human disease. The institutional problems, we obviously had the pandemic, just when I was starting to get to grips with the pathologists. And then from the genetics point of view, there was, during the pandemic, the Animal Health Trust closed, the Kennel Club Genetics Centre had to move all their frozen samples. I was very keen to make sure the Skye samples get moved, and long story short, that centre has now moved to Cambridge Vet School. Post-pandemic, the UK Kennel Club removed their funding because they were struggling financially, and the canine genetics group is now on an independent sort of foundation, but it's still the same people, it's still run by Catherine Mellish. So what we aimed to do, we sort of slightly gave up with trying to come up with a single answer, because we haven't, and we've been trying for 10 years and we haven't. So we thought the best thing to do at this stage is to describe this disease carefully, do the best we can with the genetics, and if it's not perfect, at least we'll publish it, then that will be a start, and then other bright researchers in the future will look back and say, oh, well, I know what else I can do to improve the understanding of this disease. So we've got many dogs with liver disease. I didn't count them up, but on the Animal Health Trust spreadsheet, there's a lot that have been reported as having hepatitis. But we wanted initially for the pathology just to report those where we actually have the liver histology, we actually have sections of liver. And we've got 11 dogs so far. We may have a couple more coming online before we publish. And we actually had 4 unaffected confirmed on histology as well, but there's many more unaffected. The trouble is that some of these dogs present as old as 10 years of age, which means that we can't necessarily say a dog is unaffected until late in life, but that's another story. And we have DNA from many Skyes at the genetic centres, most of them with clinical histories, some of them not. Now, in terms of the clinical presentation, the affected dogs, they do tend to be young, but not all of them. So very similar to Susan Hayward's study, we have a range of 6 months to 11 years, but more than half of the dogs were 12 months of age or younger. Now, interestingly, in these dogs, we tend to have a male predominance, and I'm a little intrigued by that. When we come onto the genetics, we don't necessarily have a male predominance in those that might be affected with one of the genes we think is involved. But we're wondering if males may be more affected by this genetic abnormality. It's definitely not sex-linked, but it may be that males are more affected, or it may just be that with a small group of dogs, this is a false result. So we can't say. Number of them were related. Most of the affected dogs were from the UK, and that's just practical, I think, in terms of getting the liver biopsies. We have one or potentially a second one from the USA who comes from a British line, and one from Finland. But I'm sure there are more. It's just easier, as I say, for us to get samples. Most of these dogs presented with this fluid in the belly, 9 out of 11. So this is a common presenting sign. The dog will be completely healthy, and then all of a sudden it'll go off its food. They'll maybe start with a bit of vomiting and diarrhoea, and there'll be an obvious fluid in the belly. 3 of them had black feces, so that's what we mean by melena, which would be typical. So having black feces and fluid in the belly would be typical of that port-like hypertension I told you about. They don't tend to have a raised body temperature. Only one of them had a raised body temperature and abdominal pain, and we think that may have been a superimposed bacterial infection, but in the majority of dogs this is not an infection. And one dog had no clinical signs, was aged 10 years and was referred, this was the dog from the US, it was referred for investigation of kidney disease to a veterinary teaching hospital, not because of its liver. The blood test findings, the elevations in liver enzymes are not dramatic, they tend to be quite mild, and as previously described, very few dogs are yellow, and an increase in bilirubin in the blood is very uncommon. So it was normal in 6 out of 7 dogs where it was measured and mildly increased in 1. One of the dogs had green teeth, so actually I say as a pup, but I went back over the notes yesterday, and it was their adult teeth that came through green, and we've had a couple of other people filling in a questionnaire that was sent around by the Canine Genetics Centre talking about this. Now that suggests jaundice in the developing youngster, which suggests that there was already a compromise in liver function, even if the dog then didn't have jaundice in later life. In dogs where it's been measured, there's been protein in the urine, so we're wondering if some of this low protein reflects kidney disease. And if you're already trying to hold lots of things in your head and thinking there's too much going on here, then join the club. There is a lot going on in these dogs, they're not straightforward. Diagnostic imaging shows the fluid, it allows us to sample the fluid, which is typical of liver disease, and the liver appeared abnormal and nodular in all dogs in which it was done. But we still need to take some kind of biopsy in order to get a diagnosis. Samples were either taken at surgery or keyhole surgery, or some of these samples that we had were post-mortem after the dogs were put down, which is very sad. Now in 6 dogs the changes seemed similar throughout the liver, but in 4 dogs they were very patchy, and some parts of the liver appeared almost normal, which may be, which again is very unusual in liver disease, may reflect differences in blood supply in utero, and may explain why some dogs do very well. So what did we find? We basically, after all these meetings, we spent 3 hours of meetings going through all of these dogs. We found 3 diseases which are intersecting, so dogs often have more than one. We found a lot of fibrosis, which is dissecting, so it's lobular dissecting hepatitis in some dogs, which has previously been reported in dogs and never in people, and is a disease of younger dogs, and nobody really knows what causes it. So I'll give you some theories in a minute. Copper was present in 7 out of 11 dogs. It wasn't typical of copper storage disease, but the dog with the largest amount had 1,000 parts per million. But we sort of agree with Susan Hayward that it may be a secondary to whatever is the cause of this disease, but it's more than you would normally get in a dog. And in at least 2 dogs, we had evidence of underdeveloped portal veins, which will have happened in utero, so what we call a ductal plate abnormality. Now, one of those dogs was the first dog we saw at Cambridge, and this set me off thinking that all of these dogs were going to have ductal plate abnormality. But we're not, we can't really definitively diagnose that in all cases, only in 2, but it's definitely there. So how are we going to tie all this together? I'll just show you a picture of the portal trial, just to show you what I mean by ductal plate abnormality. So this is the portal vein, this is where the blood comes into the liver, and then it goes down the little sinusoids here and disappears down the vein that you can't see in the middle. And the portal vein comes in at what we call the portal triad, because it's not just the portal vein, it's also the bile ducts, and it's also the hepatic artery here with a thick wall. So this is bile duct, this is artery. And these are all branches of the portal vein because it's been tortuous where we took the section. Now, some dogs are born without these terminal branches of the portal veins, so they don't develop properly. And that is like a pollard willow. So where normally the veins would all branch in your liver like a proper willow tree, in these dogs you have a pollard effect where they're cut off during development. They basically never develop the terminal branches. And that will obviously increase the pressure, back pressure from the liver into the portal circulation, because the blood can't get through. And in fact what you see lots of arteries and bile ducts but no portal veins. I haven't got a picture of that to show you. I have got a picture of patchy disease. So this is one of our dogs. These are 4 sections of this same dog's liver, And this is using Sirius Red, which shows the fibrous tissue. And what you can see, this top right-hand one, you can see lots of big nodules and then fibrous tissue between. This is typical of nodular cirrhotic changes. You know, this is cirrhosis towards the end of liver disease. And the same with this one at the bottom, which you can see is overlaid here, has a great big nodule. But this one at the top left, just one little nodule, and this one at the bottom, just one little nodule, and the rest of the liver actually is histologically almost normal. So this is very unusual. You don't normally see this in chronic hepatitis in people or in dogs. Normally it would be the same throughout the liver. So the human pathologist, this and the lobular dissecting hepatitis were the two things that surprised her the most. This is just to demonstrate to you a dog that has quite a lot of copper. All of these little red granules are copper in the liver cells. And these, this is stained with rhodonine stain. We do need to do something about this if we see it. We would typically put them on a low copper diet and maybe also give them a chelator for a period of time if it's really bad, because this copper is an oxidizing toxin. It is quite damaging to the hepatocytes, so it's better to get rid of it if we can, even if it's secondary. Don't like seeing that in the liver. Now, a key point about the liver, now you've seen the patchiness, is that it has an incredibly good reserve capacity. So both the function and the structure, it can regenerate itself, and even if you've not got much liver left, the liver can functionally behave as normal. And there's an estimate that with chronic liver disease, you need to lose 75 to 80% of your liver function before you show signs. So this is why some of these dogs are doing well, because as long as they still have 20 to 25% of their liver is normal, then they can do well. So the patchiness of the disease is a big thing. Now let's just move on to talk about the genetics, which is quite short. So 5 out of 9 cases were euthanized on first presentation, and 1 dog within 10 days of presentation because of their marked clinical signs and suspected poor prognosis. Then 1 young dog died at home 2 months after starting treatment, and 1 11-year-old dog died 4 months after starting treatment. Both of these dogs were put on immunosuppressives, and we don't think these are necessary, and it's possible that these could make the symptoms worse in some dogs, so that was interesting. Now, the remaining 3 dogs had symptomatic treatment. So we had the diuretics. I don't know why this said 5 out of 9, it should be 5 out of 11, so that's a mistake when you're looking at this. Ursodeoxycholic acid, S-adenosine, which is an antioxidant, and one of them had intermittent antibiotics and long-term antacids, phenysteine. Now, these dogs did reasonably well. One of them didn't do so well. It was put down after 8 months because of clinical signs, but one was euthanized after 2 years, age 6 years of age, and one was still doing well. We lost a follow-up after 7 years, age 7 years, 11 months. So I would say that we don't know the ideal treatment. At the moment, we would use supportive treatment. There's no evidence that immune suppression helps, and it might make the situation worse. The outcome is quite variable, but dogs can present quite sick and do really well. And so if you have an affected dog, it's definitely worth trying treatment, because some of those dogs will do well, because you can't see, you can't take a biopsy of the whole liver, so you can't see which bits of liver are still working, and remember that you only need 25 to 30% of liver function. So finally, the bit you've all been waiting for, which is the genetics, which is helpful and not helpful all at the same time. So when we looked at the pedigrees of affected dogs, initial pedigree analysis suggested that we might be dealing with a simple recessive gene. This and our suggestion that this might be an inherited or a congenital ductal plate abnormality and abnormality in liver development made us hopeful that we might be about to crack this and find out what the gene is. But it didn't turn out to be that simple. So what the Canine Genetics Group did was they did a genome-wide association analysis, which using 13 confirmed cases and 35 Skye Terriers without the disease, they picked up an area of interest, and then they whole genome sequenced 2 cases in order to see if that area of interest was interesting. And then they compared this to, at the time, 293 dogs of other breeds to see if any of them had that abnormality. And what we found was a gain-of-function mutation. I'm not telling you the name of the protein really until we've published it because it is completely novel. It was a gain-of-function mutation in a protein that is involved in transport of molecules across cell membranes. And there were some problems with this. We know that this mutation is actually implicated it's rare, but it's seen in humans, and it affects the handling of some drugs, particularly drugs used for type 2 diabetes in humans, so not drugs that we use in dogs. Now the problems with this, well, it's a protein that isn't expressed in the liver of adult dogs, it's expressed in the kidneys, and it's also expressed in the gut and placenta, which I think are interesting. The one thing that really disappointed us was that we have one clearly affected dog in our 11 dogs, and it's definitely affected, and we've checked its pathology, and we've checked everything about it, and we've even sequenced the genetics from the pathology block to make sure we haven't made a mistake. And this dog is homozygous wild type. Furthermore, what is odd, and it's not a recessive gene, is that dogs can be unaffected with one copy of the gene, and they can be affected with one copy of the gene or with 2 copies of the gene. So this is not a simple autosomal recessive inheritance. So we've come to the conclusion that this is likely a disease that has more than one gene involved, and we've hopefully picked up one of the genes. We don't know how important this is for the inheritance of this condition. We believe the current gene, because you can have one copy of the gene and either have the disease or not have the disease, this suggests that this is a dominant gene with variable expression. So what that means is that the gene interacts in some way with the environment. And since it's a transport protein, one would suggest that it would interact potentially with either food substances or drugs, or even this transport protein even transports molecules in normal animals like dopamine. So it's potentially interacting with the environment. Now my current theory, and this may turn out wrong, for these 3 different diseases that we're seeing overlapping, is that this gene may have an effect both in utero, so when the pup is developing in the uterus, and also when it's growing and when it's an adult. So you could, I've been down so many rabbit holes, you could think of so many things that could affect that could affect them. So for example, we know that smoking affects the development, smoking in the environment affects the development of babies. Could it be something like smoke? Could it be something like drugs? Could it be something dietary? Or very importantly, is it the blood supply? Is it the smallest puppy in a litter that's more likely to get this because they have an abnormal blood supply in some parts of the liver, like we saw in that Glasgow dog? So wait and see. I think the time we're doing some more studies, we're looking at, interestingly, this mutation so far we have not found in a single dog of another breed, only in Skye Terriers, which is in itself very interesting because that's again quite unusual. That's a bit like the COMD1 mutation in Bedlington Terriers that you don't find in any other breeds. So the geneticists now are back in action. They're just going to polish this by looking at more dogs of other breeds to see if they can see this. And once we've got all of the results, I hope that we'll be able to release our advice as to what to do with this. If the canine genetics group believe this is an important gene for Skye Terriers, then they are at least in the position that they can then develop a test, because that's what they do. But I'm not sure that right now I can say to you, this is going to get rid of the disease in Skye Terriers, but it may be a gene to breed out of the group. So I may have overrun, and I'm sorry about that, by 5 minutes, but just to conclude, there's so much to talk about. This form of disease in the breed. It might be associated with mutation in transport protein, but it's not a perfect association, but this may be one of the things that is causing it. I meant to mention actually that this transport protein has twice the activity. It works twice as well in entire males compared with neutered males and females. So it could explain, this in people anyway, it could explain a difference between sexes. I'm intrigued as to whether it's the genetic status of the mother, for example, that might be more important than the puppies, at least for development in utero. This disease is likely to involve more than one gene and factors in the environment, but potentially really identifying and publishing about this in Skye Terriers may help other breeds as well. For example, standard poodles have been reported with lobular dissecting hepatitis, so maybe we can help not only Skye but also other breeds. But I think listening to what I've been telling you, you can see why this has taken so long, because this is a really difficult nut to crack. There's more work needed, but given that this transport protein is also expressed in the kidneys, and given that congenital ductal plate abnormalities tend to affect both the kidneys and the liver and sometimes the pancreas, it may be that this is related to the renal dysplasia in the breed as well, but we don't know. So what to do if you have an affected dog, and I'm really nearly finished. I had a chat with somebody a few days ago who said they thought that there were no longer dogs out there, and then literally yesterday I had an advice email from somebody working in a referral practice in the UK who is treating a 5-year-old Skye Terrier that sounds as if it has this. So I'm sure they're still out there. If you have an affected dog, stay positive. Your dog may well do very well. If possible, please contact the Canine Genetics Centre for cheek swabs to get some genetic material from your dog. And while I'm here, I remembered yesterday that really those samples were mostly collected 10 years ago. If you know that you sent genetic samples from a dog to the Animal Health Trust and its disease status changed, so if you said it was unaffected and then subsequent to that, even in the last few years, it's developed liver disease, please do let us know and email Brian on that email address to let him know. He's still the same guy that was taking the samples 10 years ago. If your vet would like to discuss treatment, ask them to email me. And then the question is, should you breed from an affected dog? And I would say right now, no. I mean, anyway, you shouldn't be breeding from a dog that's sick with liver disease. I think if you have affected dogs, it's better either not to breed from them at all or to find a very unrelated dog to breed with. If you want to continue that line, but I'm not going to be able to tell you right now whether genetic testing for this gene will help or not, but I think that's a conversation I need to have with Catherine Mellish and Louise, who are doing the work on this, before we write the paper, because we're going to need to have that advice for you at that stage. So thank you very much, and I don't know if people have any questions.
Anyone?
And if there are any,
we are all speechless.
I was going to say, Kirsty, if there were any questions, you'd have to pass them. Yeah, there's a lot to think about there, and it is, you know, in one way I feel like we've let the breeders down, because right at the beginning we were hoping to find a single gene that you could test for and get rid of the disease. Having said that, I think this might anyway be a gene to get rid of in the breed. If it's unique to Skye Terriers and it's not a helpful mutation, it may be involved in both the kidney and liver disease. So we'll see. I think we'll need more work and we'll need to collect more samples. So that was the other thing I was going to say. You know, not even if your Skye has liver disease, if it has kidney disease, or if it's a healthy Skye, I think it would be really helpful to keep collecting genetic material at the Canine Genetics Centre. They freeze it all and it sits there, and so if we wanted then to look at more cases or look at Skyes with kidney, it would be really interesting to look at renal dysplasia cases and see if they have this mutation. So any samples that you want to send, and Brian can post out cheek swabs to actually take those samples would be helpful.
Then I would say, and this comes also from experience, if your dog dies and you don't know why, and it's like a questionable, do make the autopsy.
Yeah.
Because I had one bitch that was 8 years old, and the vet was absolutely sure that it has the Skye Terrier Hepatitis. She had been reading some of the studies, and And she was like, it's a hepatitis. And I was like, I have my doubts. And then the dog died. She tried to treat it, but it died within like 2 days or so. And it was put to sleep, and it was midsummer in Finland, which is like everything is closed. And then I said I want it to be autopsied. I want an autopsy because I want to be sure of the diagnosis. And then it was put to a plastic bag, and I took it to the pathology clinics in Helsinki. There was one guy who was like keeping the freezers and everything like going on, and he opened me the door and I gave the dog to him, and he said that okay, and then she was autopsied, and the pathologist report came. She had cancer in liver, and the liver there was no signs of hepatitis, no signs whatsoever, but it was cancer.
And that actually, Kirsty, thank you, that reminds me, because I put this in my notes, if you have a dog that you think is affected, and if the dog is well enough, it should have liver biopsies. You can take them non-invasively, true cuts or keyhole surgery, because without a liver biopsy, we don't know for sure that it's liver disease. Even in jaundice dogs, it can be due to other causes. So if you're in doubt, and if your vet thinks your dog is well enough to have a biopsy, then that would definitely be the thing to do to get a diagnosis. If you're having an autopsy and your vet takes sections of preferably liver and kidneys, because we're interested to get kidneys from affected dogs as well, or if you have a dog with renal dysplasia and you end up with an autopsy in that dog, we can still do the histology. So we need somebody else to take the sections and post them to us, but we can do the histology at Cambridge. We still have some money left in our Skye Terrier pot, not a lot, but enough to pay for that. And since we've seen a lot of cases, that might be that would be a good avenue to use as well. And then we would report back to you and your vets the results of that. I may not be physically at Cambridge anymore, but it's amazing what you can do by scanning slides in, and you can look at things remotely. That's how I've managed to do these meetings with the pathologists.
And the more we will get samples, the better chances we have to
Exactly. And if nobody's actually published anything on the renal dysplasia in these skies, and it's very tempting to think it's part of the same disease, because in human medicine the ductal plate abnormality affects people differently. So with the same mutation, some people will get abnormalities of the kidneys and some people will get abnormalities of the liver, and nobody really understands why that is, but it's probably again environmental. So there's no reason why the two shouldn't be linked, which is interesting.
Okay?
Yeah.
Thank you,
Thank
Penny.
you. Not at all.